Welcome everyone to this month's discussion on an interesting article published in the current issue of Neurogastroenterology and Motility Journal. My name is Dr. Purna Kashyap. I'm a gastroenterologist at Mayo Clinic in Rochester, Minnesota. It is my great pleasure to welcome Dr. Nick Spencer, who's a professor in the College of Medicine and Public Health at Flinders Medical Center in Australia. Dr. Spencer completed his PhD in neurophysiology in Monash University in 1998 before moving to the University of Nevada, where he spent 10 years. He returned to an academic position at Flinders University in 2007, and since there has been the chief investigator on nearly 15 National Health and Medical Research Council project grants and 3 Australian Research Council discovery grants. Dr. Spencer has published more than 150 peer-reviewed articles on autonomic neuroscience, and in 2016, he was elected president of Australasian Neurogastroenterology and Motility Association. Much of the research in his laboratory is primarily directed to understanding the neurophysiological basis of pain pathways in visceral organs and the neural control mechanisms of the enteric nervous system. Today, Dr. Spencer will discuss findings from his recent paper titled Motor Patterns in the Proximal and Distal Mouse Colon, which underlie formation and propulsion of feces. This was published in the July issue of Neurogastroenterology and Motility. Welcome, Dr. Spencer, and many congratulations on the publication in this journal. I would like to start with some background on the paper. If you can give us an overview of importance in this area, what is known, what are the gaps, and why you decided to embark on this study.
Great. Thank you very much for having me, Purna, and congratulations on the journal. Before I answer that good question, I'd like to just spend 5-10 seconds talking about the journal. I remember when it first came out. I was doing my PhD, and it was a new journal, and it's gone from strength to strength. So I'd like to just spend a minute to pay credit to Dave Grundy and Mike Sheeman and many others that started it up. It's now really a premier format to publish, and you should be proud. And this medium that you're doing is excellent. So basically, the field of colonic motility and GI, particularly gut-brain axis, is white hot at the moment, as you know. We organised with overseas colleagues a few years ago a special first meeting to discuss for the first time the relationship between studies of colonic motility in experimental animals and humans, a consensus paper which we published in Nature Reviews, I think 2 years ago. The similarities were significant, and yet many uncertainties remained. The field's now ready to make further steps in searching for the intrinsic sort of neural mechanisms and circuits that may be in common across species. So this paper is really an important addition, as it demonstrates that in mouse colon, an omnivore, as you know, has similar fundamental mechanisms preserved as in other non-omnivore species. So it really opens up a new field of more meaningful research to develop better models for human colonic motility diseases.
So in terms of specifically this study, what were your aims and what are the hypotheses you're pursuing?
Okay, so the main aim of our work was to really establish how the process of formation of solid feces and their expulsion occurs in the large intestine, in this case of a mouse. In previous years, we and others had shown in the colon there are different motor patterns. So there's not one unique propulsive motor pattern, which is often assumed, that are orchestrated by the enteric circuits. And I can't emphasize enough the importance of the mini brain. I think that's very well represented, as you well know, in diseases like Hirschsprung's, where any loss of enteric neurons leads to severe dysmotility. And really, so how these different neurally dependent motor patterns, how do they actually operate in transforming the softer content, which, as you know, arrives in the cecum from the small bowel, into more solid shaped pellets, which had not been investigated. So this work has sort of now been done for the first time. And I can perhaps move on to the next questions, which I'm guessing may involve the methodologies and why had that's been done earlier.
Yeah, great. You read my mind, because I was going to ask about what methodologies you use, because you just mentioned that there's something which has not been done. So it'll be good if you can talk a little bit about the methodologies, but also how you would envision if you had to use similar methodologies in humans, or are there ways by which you can apply your methodologies beyond just mice?
Sure, absolutely. Look, I think we're in a Renaissance era. I cannot believe in 22 postdoctoral years how much technologies have advanced in our world in the last 2 or 3 years, particularly with chemogenetics and transgenic biology. So we didn't use that in this particular study. What we did, we were very much firm believers in understanding core mechanisms. And what was really the core difference in this study was we combined high resolution video imaging with electrophysiology at multiple sites. So we used custom-made extracellular recording electrodes so we could correlate electrical activity at different regions along the gut simultaneously with intraluminal pressure and demapping. So it gave us a really good readout over a large organ, what was happening, not just single point recordings. So one of the major limitations of previous work in studying colonic motility patterns were the ways in which the patterns were elicited. The main methods to record motility of the colon isolated from an animal in vitro usually required some sort of transducers to record their forces or changes in length of the muscle. These, of course, often interfere with more physiological conditions of formation and expulsion of the content. So in recent years, we've really improved these techniques without the transducers attached to the gut. Okay, we know a lot of the patterns of motility we recorded in human 10 years ago was often actually induced by the recording apparatus. So a method was developed in now 3 different laboratories concurrently about 20 years ago, one of which was at Flinders, where we are, where we could extract the actual quantitative changes in intestinal muscle using these spatiotemporal maps. And these have really revolutionized gastrointestinal motility. Many laboratories now are doing spatiotemporal mapping. It's a bread and butter technique, which was used in this paper. But as I was saying, in this paper, we've added another powerful method by combining the mapping with the electrical activity at multiple sites.
Great.
Yeah, so that's really given us a major new window into how these patterns can evolve and differ as they progress down the bowel.
So what are the key findings using this methodology?
So we discovered that there are at least 3 different neurally dependent motor patterns that contribute to the formation of the feces into well-formed shapes. So the first mechanism essentially is the occurrence of sequential contractions of the circular muscle, starting at the boundary between the cecum and the proximal colon, that slowly propagate to about 40% along the length of the large bowel that shape the content into these pellet-shaped fecal masses. So this region represents a sort of functional transition between the proximal and distal colon. And as the content passes this functional transition, it's propelled into what we now know a better known mechanism, the self-sustaining peristalsis. So if the entire colon is full or is kept constantly distended with no content moving, then a third neural mechanism is generated that we identified that corresponds to previously the colonic motor complex. And they can propagate in either direction. We haven't understood yet the mechanism, how that directionality is encrypted in the circuits. We're doing this in human as well, in vitro, but it probably acts to separate the fecal content when it's too full. That's what we think this third pattern is doing.
Yeah. So, you just mentioned about the in vitro, and then you said you're doing it in humans and in vivo. So in some ways, how do you envision the impact of your paper on future research? And how would we try to translate these to moving to in vivo measurements and in vivo measurements in small animals or in humans?
Yeah, good question. So this work also for the first time demonstrates significant similarities of colonic neurally dependent motor patterns in an omnivore, a mouse, as I mentioned, compared with 2 other recent studies that were performed in herbivores, sort of guinea pigs and rabbits, as I mentioned. In all these species, there are significant differences in the motor patterns of the proximal and distal colon, with a shaping of the fecal material sort of occurring between the 2 extreme regions. If similar processes are present in human, then basically sort of their identification, now made easier with what we know what to expect, will affect profoundly the diagnosis of motor disorders and their treatment. So answering, I can get back to, if you like, but answering the last part, which I'm very excited to answer. It's very important, and I really like the early career scientists to listen to this. It's really important to remind the audience of the importance to understand biological mechanisms before we intend to expect to develop therapeutic drugs. It's a bit like if you're asked to repair the engine in a car that's broken down, but you've got absolutely no idea how the engine's working in the first place. And I do feel that community places a high pressure on developing cures for disease before we fully understand the cause of the disease. And the colon is one of the best examples. Unlike the heart, where we've got very, very good drugs, they're very specific and we know exactly how the heart works. There's not a lot of mechanistic studies now pumped directly into the heart. They're really therapeutic studies because the mechanisms are well known of the electrical conduction. That's not the case in the gut, particularly the colon. It's really a black box. So the organs are pretty poor. The organ, large bowel particularly, is long. It's about a metre long, and it occupies a large part of the abdomen. And it's remarkably, it's extremely poorly understood how the circuits work in what we, as you know the mini brain. And I should say that we've done some experiments recently disengaging the gut-brain axis in the large intestine using a novel technique I haven't published yet. And it's extremely compelling, the importance of the mini brain in transit. People have talked and argued for years that all the extrinsic sensory nerves that come into the gut from outside the gut are really critical for transit. Actually, they're not. They're really important for gut-brain communication and signaling to conscious levels, perhaps. But in terms of transit, they actually play a minor role. That's not published, but I just wanted one last sentence, if you don't mind. I noticed this week or last week that your relatively new US president is making sound progress in significantly increasing the NIH budget. So in terms of the future and what I'd like to see, this was a delight to read. When I was in the US in '99, NIH funding was 24th percentile. I think it's less than half of that now. And I'd love to see it to return back to those levels where really significant advances were being made, particularly now that we're in this Renaissance era with the development of such exciting new techniques to really probe into the mechanisms of what I think one of the most poorly understood organs in the body.
Absolutely. And, again, I think all of us are excited to see more investment in the space. And like you said, I think going back to even close to what the previous funding levels was would help not only support research, but also what we're losing is younger people shying away and trying to find other avenues. And so we might be losing the smartest minds because we make it so hard for them. So I think in that regard, both the field, like you said, it's not only complex, but it's also a black box. And I think the colon is a black box, and the small intestine is not any better either at this point. So I think we have similar problems when you think of it. So I think this work is really instrumental in getting us forward. And I think of this as not only just the therapeutic implications of something like this, because you mentioned you need mechanisms before you get to therapies, but even if we can use these technologies to then miniaturize and develop new diagnostics, that might help us get to the therapeutics in humans. And I think that's probably the first step before we even jump towards there.
Yeah, no, absolutely. That was very well spoken. And to the early career scientists listening, it's the positive is that there's some fantastic laboratories out there, and the gut it's now fortunately become a very favourable organ because it is so exciting and it is so poorly understood. And one thing I love to tell students is, if you're embarking on some graduate studies, you know, it's so much easier and much a greater opportunity to study an organ that's not well understood and not as heavily perhaps studied as other, you know, significant conditions where a lot of money is pumped into cancers and diabetes and so forth, which are no less important. But the field is open with the gut. And there is a very, very exciting era. And I can't do any more than encourage these younger students to get in touch with us, email us, ask what's going on, and really embrace and develop this exciting future that lies ahead of us.
So well said. Thank you so much. And so again, I'd like to thank Dr. Spencer for the excellent paper and assisting us with the podcast this month. I'd also like to thank our listeners for tuning in, and I look forward to welcoming you again next month with another exciting article. Thanks, Nick.
Thank you very much. Great to be with you.